Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Jeffrey Lieberman will present Early Detection and Prevention of Psychotic Disorders. Ready for prime time? The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, PTSD, and schizophrenia. Since 1987, the Foundation has awarded more than $394 million to fund more than 5,700 grants to more than 4,700 scientists around the world. 100% of all donor contributions for research are invested in grants to our scientists. I'm delighted to introduce Jeffrey Lieberman. Dr. Lieberman is chairman of the Department of Psychiatry at Columbia University College of Physicians and Surgeons, director of the New York State Psychiatric Institute, and psychiatrist-in-chief at New York Presbyterian Hospital. He is a member of our scientific council, received the 2006 Lieber Prize for Outstanding Achievement in Schizophrenia Research, and was a Foundation Distinguished Investigator Grantee in 2007 and 1999. Today's webinar will begin with Dr. Lieberman's presentation. This will be followed by a question and answer period. To submit your questions, please use the questions tab on the control panel on your screen. Feel free to submit your questions throughout the presentation, and following the presentation, I will ask as many of the questions as time allows. And now, I'm pleased to introduce Dr. Jeffrey Lieberman. Jeff, the floor is yours.
Thank you very much, Jeff, and it's really a pleasure to be here and speak to you on behalf of the Brain and Behavior Research Foundation, which has been an enormous boon to mental health care, psychiatric research, and people with mental illness for many decades. And under current leadership with Jeff Borenstein, the Board of Trustees and Scientific Council, it continues to do important work of supporting scientists, and particularly young scientists. So I'm happy to donate any of my time to make presentations whenever they ask. So today I'm going to talk about something which I really think is cutting edge, but it's not just cutting edge like a promissory for some distant time in the future, but something that really is in the offing and could really be game-changing for the care of people with schizophrenia and related psychotic disorders. And it has to do with a model of care that's called early detection intervention. And the question is how close is it to being ready for clinical use in prime time? So to begin, I'm going to show you my disclosures. I do a lot of work with pharma and biotech companies, but I take no money personally. My wife reminds me of this constantly. So nothing goes into my pocket or bank account, but I do have a lot of interaction with them for the purposes of advising and consulting and suggesting how compounds should be researched. So moving to the next slide, which is a picture of individuals who were asylum inmates back in the late 19th century. It was described by Emil Kraepelin as individuals who have dementia precox, as he called it, now called schizophrenia. And if we think about the history of mental illness and our treatment of it, there's really a few kind of inflection points where discoveries or advances were made that really changed the game in terms of how individuals are treated and what their prospects were therapeutically. To begin, Philippe Pinel at the end of the 18th century began to treat individuals who were mentally ill as patients with illnesses as opposed to prisoners or deviants, and he tried to treat them humanely within asylums. Flash forward to 1950, with the serendipitous discovery of chlorpromazine, the first antipsychotic drug, which ushered in the era of psychopharmacology. Subsequently, in the 1970s and 80s, there was the empirical validation of a whole array of psychosocial treatments, from psychoeducation, cognitive remediation, supported employment, assertive community treatment, which were evidence-based psychosocial treatments to support individuals with schizophrenia in a disease management model. And then I would submit to you, in the 21st century, there's been the advent of the early detection intervention model of care, which I think is going to bring all of the previous advances together in a way that could change things for all future individuals who will be vulnerable to developing schizophrenia-related psychotic disorders. So the picture that's shown in this monograph is really of the end stage of schizophrenia, which is what happens to people when they get ill, usually in their late teens or early adulthood, and there's no treatment, and their illness waxes and wanes, remits and relapses, and eventually leaves people in a way that they're persistently symptomatic and disabled. So that's the end stage of the illness. Now, the various etiologic and pathophysiologic hypotheses that have been proposed to explain what causes schizophrenia are listed here. And of course, the genetic hypothesis has been dominant, since we know that schizophrenia tends to run in families, albeit in a way that defies understanding of mode of inheritance and genetic mechanisms. And we know that it's very much polygenic. There's the dopamine and glutamate hypotheses, the two principal neurotransmitter hypotheses, which describe the neurochemical dysfunction which leads to the expression of the symptoms of schizophrenia. There's also evidence to support the idea of an autoimmune or an infectious pathogen causation of the illness, but these are kind of lesser theories in terms of the evidence. But probably the most influential of all the hypotheses in terms of explaining the development of the illness has been the neurodevelopmental hypothesis, which holds that genes or early insults to the developing fetus or at birth produces a vulnerability to develop the symptoms of schizophrenia at the pointed period of risk in the second or third decade of life. And this model has been extremely influential because we know genes are involved. We think that they affect the brain's development. And there are some telltale hints in some people of having premorbid manifestations of this vulnerability in terms of their motor function, social function, or intellectual function. However, this is not really fully explanatory in terms of what we know as the natural history and the course of the illness. And that's shown here in this slide. So we know that schizophrenia tends to have its origins early in gestation, either through genetics or through environmental insults, whether it's toxins, whether it's trauma, whether it's any kind of infectious exposure. But even if there is this damage inflicted, this vulnerability inflicted, it's pretty much dormant through childhood until after puberty. And it's only after puberty that people enter a period of risk to experience the onset of schizophrenia. And the onset doesn't occur abruptly for the most part. It usually occurs gradually or iteratively, but eventually the disturbances in the brain that produce aberrations in mental function, perception, ideation, emotion regulation crystallize into the symptoms which are diagnosable as a first break or first psychotic episode. And that which has gone before has been retrospectively or retroactively termed a prodromal phase to the illness. Now, contrary to what had been for really centuries, the notion that people who became psychotic with schizophrenia were kind of doomed to this inexorable deterioration and disability. Studies in the 1990s and the early 21st century in early phase patients showed that the response rate to treatment in first episode patients was actually quite good. A large proportion could have symptom remission and recovery. But then, since treatment guidelines did not say if you recover from a first episode, you should stay on medication indefinitely, people didn't. And they often stopped even before the continuation period of treatment was over. They objected to side effects. They didn't think that this illness was something which would be recurrent. And it's in this context of recurrent episodes that the phenomenon which led Kraepelin to first christen this illness, dementia praecox, dementia occurring early in life as opposed to senile dementia, this clinical deterioration occurred. But this progressive nature of the illness didn't kill people like occurs with classic neurodegenerative brain diseases. It produced an end stage where they're chronically symptomatic and disabled and characterized, in addition to their psychotic symptoms, by negative symptoms, cognitive impairment, and functional disability. Now, the neurodevelopmental hypothesis and all the other hypotheses fail to explain this course of the illness. And that fact begs the question of what is causing this progressive change and this progression and deterioration to the point of no return, such that individuals that experience this recurrent episodes, this intellectual decline, this persistence of symptoms, this diminution of treatment responsivity, what is underlying that? And with the advent of modern non-invasive imaging techniques like magnetic resonance imaging, what was described was the fact that if you take individuals who are early in the course of their illness and you study their brain with an MRI and compute various measures such as the volume of different tissue compartments or regions of interest, and then you follow them over time, several year period, you find that there is some progressive change in the volume of certain regions of the brain, which is particular to the gray matter. And what this movie is showing you is, in a color-coded cartoon, is that if somebody at their first episode, when they're imaged, had no change over a 4 to 5 year period when they were repeatedly re-examined, then their brains would be all blue. But when it turns more red and pink, then it's showing a change from the baseline value in the direction of declining or showing atrophy. And you can see this process occurring. Now, this process is something which occurs naturally with aging in a much more gradual way. It also occurs in other brain degenerative diseases like Alzheimer's disease. And in the case of Alzheimer's, it's a more aggressive form of atrophy. So the loss of volume in schizophrenia is more subtle, but nevertheless is discernible and is believed to underlie this progressive nature of the illness, which produces the chronic symptoms, disability, and diminished treatment response. Now, exactly what is contributing, what is actually happening at the cellular level, which is leading to this volume reduction that can be measured quantitatively with MRI. So we're looking here at a cartoon of a pyramidal cell in the cortex. This is the cell body, the axon. This is the dendritic arbor, which forms the arbor where all other input from other cells is occurring in the form of synaptic contacts. And the synaptic contacts are each reflecting a synapse where the axon terminal, once the cell is stimulated with an impulse, releases the neurotransmitter and stimulates the postsynaptic cell. Now, in the case of schizophrenia, you have a dysregulation of the trafficking of these neurotransmitters, dopamine, glutamate, which produces the symptoms, and if allowed to persist or are recurrent can potentially cause a toxicity which destroys that synapse, ablates it. And you can see here in these post-mortem photomicrographs of a dendrite with these axon terminal, these dendritic spines, which are the terminals on which the axon synapse, that in the non-mentally ill cell, you see the dendrite with all these spines here, and you see very few of them in this individual's dendrite with schizophrenia. And the inference is that the dendritic spines have been pruned as a result of the toxicity. And when this pruning occurs as a result of the fulminant pathophysiologic state of psychosis, this dendritic arbor shrinks. And it is that which is being detected morphometrically on the MRIs, which is showing the volume reduction. So the treatments we have can neurochemically modify this dysregulation, but once you lose it, it can't stimulate regrowth. And this begs the question about whether the neurodevelopmental hypothesis needs to be amended with some inclusion of a degenerative process which may occur coincident with the onset of the illness. And if that is the case, then we can, and I'm dating myself because when I was in training, there was a very pessimistic therapeutic attitude towards what could be done to forestall the deterioration that people with schizophrenia experienced. And it was, as in the words of a famous colleague of the United Kingdom, thinking that people were genetically doomed from the womb. But in the context of thinking about the course of the illness being driven by a progressive degenerative process, maybe there is the potential for modifying the course of the illness. And you'll recall that in the development efforts to develop treatments for brain diseases like Alzheimer's disease, Parkinson's disease, Huntington's disease, there are two classes of medicines. There's symptom-improving medicines like Aricept, and there's disease-modifying medicines. Well, actually, there aren't any disease-modifying medicines yet. There have been efforts with the vaccines, but none has proved effective. So the question is can we modify the course of schizophrenia in a way that has been striven for with respect to the neurologic degenerative diseases, but is yet to be attained. And that's what I'll focus on for the remainder of the talk. So are antipsychotic drugs, in the current forms that we have them, symptom improving or disease modifying? Well, we can say that based on numerous studies, absolutely positively, that antipsychotic drugs can suppress psychotic symptoms and prevent their recurrence. And we also know that they prevent various types of side effects, neurologic, metabolic, weight gain, sedation. So these are absolutely proven properties of this class of medicines. We also know what their limitations are. They can't alleviate psychotic symptoms in everybody, and there are some individuals which are wholly refractory. In addition, they have limited to no efficacy on negative symptoms and cognitive symptoms. The question is, can they, if used judiciously, prevent progression, be disease modifying? So here's some data which bears on this. These are from studies done by a colleague, René Kahn, used to be in the Netherlands at the University of Utrecht, now is at Mount Sinai in New York. And he studied a cohort of patients over 5 years in which he obtained MRI scans of their brain annually. And what he found is what's been shown in many studies now is that individuals with schizophrenia over time do have some reduction in the volume of their gray matter, which is reflected here in terms of gray matter density. Now, when he then examined within his large cohort of subjects the amount of treatment that people received during the scanning interval, what he found was, and the scanning interval was over a 5-year period, he found that the more medication people had, the less volume loss they had in terms of reduced gray matter density. Now, I just showed you the data from clozapine because it's the most dramatic, but the same slope of improvement mitigating the volume loss was seen with other antipsychotics used, and preferentially with the second generation antipsychotics relative to the first generation. So this suggested that the kind of volume change that occurred in the context of the illness was something that antipsychotic treatment mitigated to the extent that individuals were compliant and remained on treatment. This is another way of illustrating a similar finding in another study. This was a study that I was involved with where first episode patients were randomly assigned under double-blind conditions to take either haloperidol or olanzapine in the first episode and then followed for at least a year, controlled blinded treatment. And they had MRIs done at specific time intervals. And what you see here is different angles of the brain at different time points. And if there was no change in the volume of the regions of the brain from the baseline scan, the color is blue. If it changed, the color became more red. And you can see that the haloperidol subjects show more red, indicating volume loss compared to olanzapine. Now, I'm not ready to ascribe neuroprotective properties to the pharmacology of olanzapine, because this could be treatment compliance, could be the side effects cause people to stop their medicine. But clearly, there's an ability to modify the volume loss that is shown with treatment. So, if one accepts that as the possibility that there is, one, a progressive nature to the illness, in addition to the neurodevelopmental diathesis from which the illness springs, and that treatment might be able to modify that if administered properly, then what are the real kind of pressure points or points of vulnerability that need to be addressed therapeutically in order to be able to deliver on this potential for disease modification? Well, the first thing is limiting the duration of untreated psychosis, how long people are actively symptomatic before they come to treatment. And the second is maintaining people in treatment and preventing any recurrences in the form of psychotic episodes and relapses. Well, 2 studies were done that were pivotal in testing these hypotheses. The first was called the TIP study. It was done in Scandinavia. One of the lead investigators was Tom McLashan from Yale. And this study was testing the feasibility of being able to reduce duration of untreated psychosis and its relationship to outcome. And what they did is they did a geographic study where they assigned regions in Denmark and Sweden to a new kind of intensive, comprehensive early detection, which involved public service announcements, increasing public awareness, outreach, and they compared it to usual dissection strategies, letting people get sick and come for treatment whenever they felt the need to. That was done in Norway. And then treatment in both places was the same as usual. And what they found was that the duration of untreated psychosis in the intensive region was reduced from 26 weeks to 5 weeks, and the usual in the treatment as usual region was 16 weeks. But most importantly, when they examined outcomes at various time points, they found that the outcomes in the intensive regions were better of subjects who had a reduced level of duration of untreated symptoms before their first treatment exposure. Since then, there have been numerous studies which have related DUP to prognosis, all in the same direction of longer DUP, less likelihood of recovery, a longer time to recovery, less degree of symptom remission. The second question about once you get patients into treatment, how do you keep them in treatment, keep them engaged, not allow them to discontinue even if they don't want to take medication, but to keep them engaged in some therapeutic connection. And this was addressed in the context of an NIMH-funded RAISE study, recovery after an initial schizophrenia episode. And what this was trying to do was basically to see if a team-based, coordinated specialty model of disease management, an optimized intervention for first episode psychosis, would facilitate symptom remission and recovery, prevent illness progression, and limit disability. And the specific elements of this optimized intervention were called coordinated specialty care, involved medical management and psychopharmacology, but also psychoeducation, case management, rehabilitation, substance abuse treatment, suicide prevention. And not everybody received all of these, but a determination was made based on an individual care plan of which of these that they needed. But basically, this is trying to provide all available treatments that are shown to be effective in managing people with schizophrenia and providing them right up front at the beginning of the illness. But also, very importantly, the philosophy of this model is that there is a shared decision-making, engaging the patient and making them feel like they're part of determining what their care plan should be, and that it's not a unilateral, authority-driven type of treatment model. And it shows respect for the patient's point of view and also acknowledges the tenets of the recovery movement. Well, here's just some data which illustrate the benefits of this approach. So these are data from one of the RAISE studies that Lisa Dixon oversaw, and you see that, look at this rate of remission and the sustaining it, and the global assessment of function score over time. So this is not following patients for decades, but is following patients for some number of years and showing this continuity, which the assumption is if that can be sustained, will then forestall the deterioration that's traditionally associated with the illness. So, based on the body of work that culminated in those studies, my opinion is that for syndromally diagnosable psychotic disorders, that early detection intervention in this coordinated specialty care model is ready for prime time and should be the standard of care. It's not everywhere, but it should be. And it depends on the infrastructure, the financing through CMS and whatever third-party payers there are, but this should be the standard of care available in all locations. Now, never being satisfied, which we shouldn't be until we've eradicated this dread illness, researchers began thinking, well, if we can intervene when people are diagnosable and stop the illness, or at least slow it in its tracks, since we know that the illness has this prodromal period, what if we try and identify people sooner? So we can prevent progression by treating people optimally in the first episode. What about the prodrome? So makes good sense, very, very attractive idea. It also reflects Ben Franklin's adage, an ounce of prevention is worth a pound of cure. But the question is what is the methodology? What is the actual way in which this concept can be applied, and is it ready for prime time? So the early detection and intervention strategy applied to individuals pre-syndromally looks like this. Mostly it's related to help-seeking people. It's not like community-based surveys are looking for people who may have prodromal symptoms. It's people who come seeking help for symptoms that don't meet DSM criteria for a diagnosable disorder. And they're assessed and they are characterized as meeting criteria and as being at clinical high risk. Now, here's the first problem, is that the different criteria that have been developed by researchers around the world are different, and each has different limitations. So blip refers to a brief, limited, intermittent psychotic episode, and it means that someone has some psychotic symptoms, but at a low frequency and duration, and they don't meet syndromal criteria, and they don't last very long. This is attenuated psychotic symptoms, and it's really now probably the dominant criteria that are used for identifying people at clinical high risk for psychosis. They have attenuated symptoms. There's sub-threshold positive symptoms like unusual ideas or suspiciousness, perceptual disturbance, but they don't have psychotic conviction. They're not full-blown. This is a European group in Switzerland that were called unspecific prodromal symptoms. It's not very precisely defined and not widely used. This refers to basic symptoms, and it's another kind of variation on these sort of nondescript, non-specific symptoms that are dysphoric and unpleasant and represent change, but aren't necessarily characteristic of psychosis. And this is genetic risk and deterioration, which means that an individual comes in and they're complaining of different types of non-specific emotional or ideational symptoms, and they have a first-degree family member with a psychotic disorder. And, importantly, they've had a functional decline which have occurred within the past year. Drop off in their grades in school, not being able to perform at work, withdrawing from social function. So these are the different criteria that are used to define people who are to be at clinical high risk. And then if they are included in this group as bearing some type of monitoring or symptomatic treatment, they're followed, and they're followed for some period of time to see if they do convert to a syndromal psychosis or not, and they either normalize or just continue stably with these low-level subclinical symptoms. So that's the methodology that exists based on clinical criteria, which is really all that we have to make assessments of who's in the so-called prodrome. Now, here's the problem. This shows you, applying these different sets of criteria, what the transition rate was to a DSM syndromal psychotic disorder. So this first study was by the Australian group of Pat McGorry and Alison Young, and they initially reported in these clinical high risk that about 40 to 45% of them converted over several years. But as you can see, the rate is lower and lower and lower and lower. And what this means is that although these individuals went over this period of time from having this clinical high risk, quasi-prodromal set of symptoms, that the majority of them never converted to a syndromal disorder, meaning that you have false positive rates of upwards of 70%. And that's not good. So in order to be able to say you can diagnose an illness or a group that are at risk for that illness, you have to meet certain requirements in terms of the rigor of the diagnostic criteria. And these are quantified in terms of their specificity and sensitivity, and in terms of what's called positive predictive value and negative predictive value. So the sensitivity means that your criteria identify everybody or most people who have these prodromal signs who are going to convert to a syndromal psychosis. However, you can have high sensitivity and low specificity. That's saying you get everybody who converts, but you also get a lot of people who don't convert. And you have a low specificity, which means that you have a high false positive rate. A positive predictive value is being able to say that you're able to identify all of the people that all of the people that are going to develop the illness, and the negatives that you're going to be able to identify with high certainty, all of those that are not going to develop the illness. So if we then look at the different criteria that have been published, and I'm just showing two of the criteria for psychiatric symptoms, you can see that the sensitivity, the specificity, the positive predictive value, the negative predictive value are not all close to 100% or 90% or high enough so that they have an acceptable limitation. Now, you compare these to the Framingham criteria for cardiovascular disease or mammography for breast cancer or the PSA, and you see that this is a situation that exists for a lot of criteria. And for PSAs, you have individuals that have a high specificity but a low sensitivity, and low positive predictive value, high negative predictive value. So this is something that is an issue that exists for all of the, the various calculator or risk tables of risk factors that have been quantified into these nomograms to predict vulnerability to illness. And we're experiencing the same with our criteria for incipient psychotic disorders also. So this is a good start, but as to whether it's ready for prime time, that's an open question, because the risk is that you're assigning a diagnosis or the possibility of a diagnosis with all of the stigma and psychological consequences to individuals who may not warrant it. And if you're using any kind of treatment to treat them, you may be assigning them to a treatment that they don't actually need. So, and this issue about whether this set of criteria, the pre-syndromal form of schizophrenia, was ready for prime time, was debated
by the DSM V steering committee, and they considered whether they should include in the DSM V an attenuated psychotic syndrome or a clinical high-risk syndrome, and they decided that it was not ready for prime time, which is why it was put into the appendix. Well, the idea is a good one. There still needs to be work on the case identification criteria process, but that didn't stop researchers from trying to go on to the next question, which is, okay, if we identify people that we think are at risk, what can we do to treat them that would not just alleviate their prodromal symptoms, but also prevent the onset of the syndromal psychosis? And there have been a number of published studies, although this issue is really far from clear. The first and third studied antipsychotics, and the results here are, I would say, mixed. The cognitive behavioral therapy was not also informative, in my opinion. There was a study which was highly positive in an almost, results too good to be true fashion, which randomized prodromal subjects to omega-3 fatty acid or placebo, but this study was never replicated, so it was too good to be true. The current, I think, leading candidate for a non-antipsychotic drug approach involves drugs that modify glutamatergic neurotransmission, and I'll show you some data on this in a minute. So, what is needed to make this strategy in prodromal patients ready for prime time? Well, we need to improve the diagnostic specificity and precision, and we need to determine what the best therapeutic approach would be, other than monitoring or just symptomatic treatment. So our group has been focusing on this, as have many others. We decided to determine where in the brain we should look to identify a biomarker that might validate the clinical criteria and improve their specificity and sensitivity. And of the regions of the brain, we found in a review of imaging studies that was done that the hippocampus was the area that was most commonly affected in terms of the different ways in which brain structure and function are studied in schizophrenia. And there's a lot of reason to implicate this hippocampus in the early stages of the illness. So we focused there. And what we did was we developed an imaging technique that measured function, reflecting metabolism in the hippocampus, which, and this is a coronal section through the hippocampus. Remember the seahorse shape of the structure, which has multiple cytoarchitectural neuronal fields, and could distinguish the different subfields, because we felt that in order to be able to have a biomarker diagnostic specificity, we need to have spatial resolution, which could define the different regions, subregions within the hippocampus. And using this imaging technique, we examined individuals with schizophrenia, healthy volunteers, and then individuals in the clinical high-risk state. And what we found was that in the schizophrenia individuals, there was, relative to controls, this hyperactivity in the CA1 region of the hippocampus, also extending towards the subiculum. In the prodromals, we also found this to a lesser degree in the CA1, and that this elevated metabolic activity measured by the blood flow and metabolism was associated with the symptoms, their positive symptoms, and was distinct from that found in the controls. So this hotspot seemed to be a marker for the earliest stage of the illness. Interestingly, we followed individuals over a 2-year period, and there was in our sample, which now is up to an N of 200, a conversion rate of 30+% to syndromal psychosis within 2 years. And whereas at their baseline assessment, there was no marked change in the hippocampal volume, that by the time they converted to psychosis, there was. And this is just a representation of the surface of the hippocampus, which is shown here in coronal section on MRI, showing the area where there was a volume change. And this area where there is volume change corresponds to the area of the greatest levels of metabolic activity. So atrophy or volume change followed the elevation in metabolic activity, which we believe is reflecting a neuronal activation sequentially. This is another way of looking at it. So here we're looking at the subiculum, the CA1 region, which show the volume change. And this is time 1, time 2, from the baseline to when they converted, or 2 years. And you see that this is the CA1 region. And in the converters, it was elevated, but then stayed relatively flat. In the subiculum, it increased, showing the spread. And here we see the volume of the hippocampus in this region, showing the reduction. So the question then is, what is causing this elevated metabolic activity and subsequent volume change? And for a variety of reasons, we thought it had something to do with glutamate-mediated neurons that were in this region. And this is a cartoon of the glutamate synapse. Here's the presynaptic axon, postsynaptic cell. This is the synaptic vesicles. This is the NMDA receptor, whereas different constituent parts. This is the astrocyte, which absorbs and recycles the glutamate. And there's different ways to modulate the synapse. The ones that have been used most commonly have been the allosteric modulators like serine or glycine transport inhibitors. But there's other ways to do it also, including by stimulating these mGlu-R metabotropic receptors, which can inhibit the release of presynaptic glutamate. Now, you can measure this in vivo in humans using magnetic resonance spectroscopy. You define a voxel in a region of interest. This is the frontal cortex, but could just as well be the hippocampus. You get a readout like this, which shows you the spectrum, and these resonance peaks equate to a concentration of these biochemical constituents, choline, creatine, N-acetylaspartate, and GLX is a combination of glutamate and glutamine. And when you look at the MRS spectra and look at the GLX peak, you see this activity where there's increased levels in the individuals who convert compared to non-converters compared to controls, suggesting higher levels of glutamate in that voxel or in that region of interest. So we then wanted to run an experiment to see could we modulate that with pharmacologic agents, and of the ones that we could use, we selected the mGlu-R partial agonist to inhibit presynaptic release. And to test this, we first studied an animal model where we took mice, we injected them with ketamine. We had a small animal magnet where we could measure their metabolic activity in the hippocampus, and we found that ketamine produced this identical pattern of subicular and CA1 elevation in metabolic activity relative to mimicking what we saw in the human subjects. And then we ran the experiment where we put a dialysis probe in that part of the brain and measured, after ketamine was administered, the level of glutamate in the synapses of those cells. And here you see the curve for the CA1 region. This is the entorhinal cortex, and you see this spike showing the increased surge in glutamate after ketamine. And it corresponded to these regions that we had seen and that ultimately are subject to atrophy with repeated administration of ketamine. But then if you pre-treat the animal with this drug, which is a mGlu-R 2,3 partial agonist, pomaglumitad, you inhibit the spike in metabolic activity that is produced by ketamine. And because we extended the experiment, repeating the ketamine administration over 30 days, if you pre-treat, you also can prevent the atrophy from occurring in these animals. So right now we have an experiment ongoing where we're doing this in patients, and the expectation is that we not only could alleviate their immediate symptoms, but this has the potential to forestall the onset of the illness. And this really reflects a model of pathogenesis in which individuals go from a premorbid state to a state where they enter a period of clinical high risk, which begins with dysregulation of glutamatergic transmission. This results in increased neuronal activity, which affects metabolism and blood flow, and ultimately can lead to atrophy. And this parallels the model that is seen in Alzheimer's disease. So here's our model for the onset of schizophrenia. With Alzheimer's, which neurobiologically has been characterized more extensively, you see that in elderly individuals who have no obvious cognitive impairment, there is the beginning of beta amyloid accumulation that can be detected with PET scanning. But then tau-mediated neuronal injury occurs subsequently, and then you get the consequences in terms of atrophy as a result of the toxic protein aggregation. And this then affects memory and then overall clinical function. So we think that this pattern may sort of simulate the pattern, albeit via different pathogenic mechanisms as occurs in dementia. So dementia precox and senile dementia may have a similarity in that regard, albeit with different pathophysiologic constituents. So At this point, we don't have regenerative treatments, but we do have the capacity to prevent Humpty Dumpty from falling off the wall. But you have to get there in time, and you have to administer the treatment in the right way. Ultimately, I hope we will have regenerative treatments, but there's really none on the horizon. So we really need to focus on that early phase and heed Ben Franklin's dictum about an ounce of prevention. So the state of the art that exists for first episode is here. We aspire to this, but need to improve our methodology in terms of case identification and treatment. But clearly, it is something that is within our grasp and that is realistically achievable. So to conclude, schizophrenia is a progressive disorder in addition to its evolution from a developmental diathesis. Clinical and hopefully soon biological measures will allow us to identify people at risk. And if we do so, we do have the potential to intervene in a way that can modify the course of the illness and minimize the morbidity and disability that historically has occurred in relation to this illness. And right now, I feel very confident saying that early detection intervention is ready for prime time with DSM diagnosable psychotic disorders and is on the horizon for intervention in the pre-syndromal, prodromal stages. But first we need to improve the case identification diagnostic criteria and determine what are the optimal ways of intervention. So let me just stop by saying that the work, at least the data that I showed you from our program, is reflected by individuals who were the key persons here. And in your spare time, if you want to read something that's better than Ambien, I published a book. This is an immodest, self-serving advertisement. And thank you very much for your attention. And if we have any time, Dr. Bornstein will field some questions.
Yes. Jeff, thank you for, first of all, for the work that you do and for really looking at a complicated subject and presenting it in a very user-friendly, lay-friendly way. So I appreciate it. A question that's come across from a couple of people, I think there are people listening to this who may be concerned that their loved one, their child, may be in this prodromal phase. What should you do? You're concerned. What's the next step for a family member?
I always say that you err on the side of caution and seek help. And so you should go see a psychiatrist. And a lot of people say, well, should I see a social worker, see a therapist, psychologist, I see my clergyman? I think you should start with the individual that has the highest level of training. If you can't get to one, then the next best, whether it's a nurse practitioner or whether it's a psychologist, but somebody who's skilled in treating mental illness as opposed to the worried well. Then there needs to be an evaluation as to whether this is just, a phase somebody is going through, whether this is basically one of the kind of transient maturational stages that youth often go through, or whether it really does seem to be kind of the precursor to a mental disorder. And it may not be immediately apparent, but that's okay. You don't have to rush to do anything. It just means that they should be followed over some period of time to see how it evolves. And treatment, to the extent that treatment is administered with an uncertain diagnosis, is treatment just for symptoms. So what I mean by that is if in the context of an individual having changes in their behavior and lifestyle, they're also feeling anxious, or they're also having sleep problems, or they're depressed. You can use an anxiolytic. You can use sleep hygiene. You can use an antidepressant. You can treat symptomatically while you're following and waiting to see if the condition evolves into something more serious. So err on the side of caution. Don't wait. I have heard so many people saying, if I only had known, if I only had known. There's nothing to lose except the time and whatever the copay is to go see somebody.
I think that's an important message, because when we look at the data that you presented earlier about the delay in somebody getting treatment once they become more fully ill, by even going during this questionable prodromal phase, you're now on the radar, so that you're looking to see that maybe symptoms are developing and can get treatment sooner rather than later and nip it in the bud if it does go on to a full psychosis. And that can make a big difference for the person as well.
And there's also, Jeff, one other complicating factor in this. Oftentimes when individuals begin to exhibit these suspicious behaviors or symptoms, they often don't see them as being a problem or see them as being something unusual, different, or signs of illness. And they may not want to go to see somebody. So a family member has to be quite careful and strategic in how they get them to see a mental health professional. But again, I encourage people to engage in a little bit of coaxing and a little bit of imaginative approaches, like, I'm having difficulty in the way that we're sort of relating to each other, and I think it would be good if we could go talk to somebody. So it's important, I think, to recognize the fact that it's not like, I just twisted my ankle and I have to go see an orthopedist, and that everybody will agree that that's the course of action. In the case of mental conditions, that's not something that everybody recognizes necessarily and feels as part of a possible illness or something wrong that may need professional attention.
The very important point, both for this prodromal phase and down the road, of how to engage people, encourage people to seek help.
Oh, and Jeff, there's one other thing also. I have to, with all of this, legalization or decriminalization of cannabis products.
You just read my mind. That was the question I was going to ask you. You just read my mind. Go ahead.
No, go ahead.
Yeah, no, I was going to ask you about exactly what you're saying about the marijuana and the other cannabis products. Tell me about your concerns and thoughts about that.
Well, recreational, intoxicants are used, pretty broadly by youth in our culture, and now with cannabis being decriminalized and legalized, it's becoming more so, and the forms in which it's administered and the strength is increasing, and there's no question it is a contributing factor to precipitating psychoses. Now, we don't think that drug use by itself causes persistent psychotic disorders, that it simply precipitates or accelerates the onset in vulnerable individuals. But frankly, we're not completely sure. But there's no question that individuals who have some predisposition, if they use cannabis products particularly frequently and heavily, are at substantially greater risk. And what that means is that even if they stop using it at some point, they may have already caused the illness to arise, and then basically, you have to treat a persistent illness. So that's something that's very important for parents to observe in their children.
Good. Thank you for making that point. Our time is just about up. So, Jeff, I want to again say thank you for an outstanding presentation and for all that you do. I also want to thank everybody who joined us today. All of the research we fund is made possible through private donations. So please consider making a contribution by visiting our website, bbrfoundation.org, or calling us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with a family member or a friend, please visit the events and webinar page on our website. I hope you'll join us again next month when Dr. Eric Nessler, Dean for Academic and Scientific Affairs, Director of the Friedman Brain Institute, and Professor of Neuroscience, Pharmacologic Science, and Psychiatry at the Icahn School of Medicine at Mount Sinai, will present The Biology of Addiction. This webinar will take place on Tuesday, March 12th at 2 p.m. Eastern Time. Thank you again for joining us, and enjoy the rest of your day. Take care.